Close your eyes and picture a forest. You probably see trunks rising from the ground, branches spreading overhead, leaves filtering sunlight. What you're picturing is accurate, but incomplete. A forest doesn't spread horizontally like a carpet. It builds vertically, stacking life in layers from soil to sky, creating distinct worlds separated by mere meters, but living under entirely different rules. Stand at the base of an old-growth rainforest and look up. 200 feet above you, individual trees punch through the main ceiling of leaves into open sky. These emergent giants can reach heights of 60 to 80 meters, their crowns isolated from neighbors, each standing alone like an island in a sea of green. They endure the full force of weather, scorching sun, battering wind, direct lightning strikes. Life at this height demands specific adaptations. Emergent trees produce small, tough leaves with waxy coatings that minimize water loss. They spread crowns wide, maximizing exposure to unfiltered sunlight. Many produce winged seeds that catch wind and carry far, avoiding competition. The animals here must navigate extreme exposure and scattered footholds. Eagles patrol these heights, their massive wingspans allowing them to soar between isolated crowns. The harpy eagle, one of the largest raptors on Earth, hunts from emergent perches, its wingspan reaching 7 feet. Pygmy gliders navigate the emergent layer using skin flaps stretched between their legs, gliding between trees under darkness. Descend 40 feet, and you pass into a different world. The canopy begins here, a continuous roof of interlocking branches forming a layer roughly 6 meters thick. This is where the forest truly lives. The canopy houses roughly 50% of all plant species, and possibly a quarter of all insect species on Earth. The architecture is dense and three-dimensional. Tree crowns pressed together, branches weaving into a nearly solid platform. Vines loop between branches, creating aerial highways. Epiphytes, plants growing on other plants without parasitizing them, festoon every surface. Orchids, bromeliads, and ferns cling to bark, their roots dangling, absorbing moisture from rain and fog. Some collect rainwater in leaf tanks, forming tiny aquatic habitats where frog species raise tadpoles. The canopy blocks wind, rainfall, and sunlight, creating humid, still, dark conditions below. It intercepts most rain, channeling water down trunks and dripping from leaf edges. It dampens temperature swings, creating stable conditions, and critically, it filters light. Life concentrates here because of light. Photosynthesis happens predominantly in the canopy, where leaves access direct sunlight. This energy capture feeds not just canopy dwellers but the entire forest below through the constant rain of leaves, branches, fruit, and waste falling downward. Animal life in the canopy exceeds that of any other layer. Monkeys swing through branches, traveling miles without touching ground. Scarlet macaws and toucans use oversized beaks to crack hard-shelled fruits. Sloths move slowly, eating leaves and sleeping in high branches. Insects here live entire life cycles without touching earth. Ants build colonies in hollow branches. Beetles bore through bark. Butterflies flutter in dappled light. The biomass of canopy insects likely exceeds all other forest layers combined. For decades, the canopy remained largely unexplored. Scientists on the ground could observe the forest floor easily, but the canopy stayed out of reach. In 1917, naturalist William Beebe wrote that another continent of life remained to be discovered, not on Earth, but 1 to 200 feet above it. Methods developed in the 1980s, rope climbing systems, canopy cranes, walkways, even hot air balloons, finally gave researchers access to this aerial realm. Drop another 30 feet, and light dims dramatically. Only 5% of sunlight reaching the canopy penetrates to the understory. This layer exists in permanent shade where only specialized plants survive. Plants here have adapted visibly. Leaves grow larger, maximizing surface area to capture scattered light. Some produce red or purple pigments that help increase photosynthetic efficiency by reflecting additional light back through leaf tissue, giving photons a second chance at absorption. Many understory plants produce intensely colored, strongly scented flowers, bright reds, deep purples, vivid yellows. In dim light, pollinators navigate more by scent than sight. Animals of the understory operate in twilight with senses tuned to low light and high humidity. Jaguars patrol tropical forests, their spotted coats breaking up their outline in dappled shadows. Tree frogs navigate between leaves, their bright colors warning predators of toxicity. Snakes drape over branches, waiting motionless for prey. Birds fill the understory with sound. In the absence of clear sightlines, vocal communication becomes critical. Many species have developed elaborate songs and calls to establish territories and attract mates through dense vegetation. The final layer, the forest floor, receives only 2% of sunlight hitting the canopy. Walk through old-growth forest, and you might be surprised by how open the floor is. Popular imagination pictures jungles as impenetrable tangles, but that image applies to forest edges where light reaches the ground. In deep forest beneath intact canopy, the floor remains relatively clear because almost nothing can photosynthesize in such darkness. What does thrive are decomposers. The forest floor is carpeted with fallen leaves, dropped branches, dead insects, shed bark, animal waste. Fungi bloom from this debris. Bacteria work invisibly, breaking bonds, releasing nutrients. Insects burrow through litter, chewing and digesting, transforming dead matter into simpler compounds. Decomposition is rapid in tropical forests, where heat and humidity accelerate decay. A fallen leaf might break down in weeks. In temperate forests, the process slows. Leaves fallen in autumn might persist through winter, but everywhere, the outcome is the same. Nutrients locked in dead tissue get released back into soil where roots absorb them, completing the cycle. The floor is also where most seeds land and germinate. Saplings crowd the dim spaces between mature trees, waiting. Most will die without reaching the canopy, but when a gap opens above, when an old tree falls or lightning strikes, light suddenly floods a patch. The waiting saplings race upward, competing to claim the opening. Only 1 or 2 will succeed, eventually filling the gap and restoring canopy continuity. These layers aren't sealed off. They interact constantly. Rain drips from canopy to understory to floor. Animals deposit seeds through droppings. Nutrients absorbed by roots travel up to canopy leaves, then fall back when those leaves die. Energy captured by canopy photosynthesis flows downward through falling fruit, feeding ground-dwelling animals, whose waste feeds decomposers, whose activity enriches soil, supporting roots that feed trees, making up the canopy. But each layer maintains its own distinct climate. Temperature, humidity, light, and wind speed all change as you move vertically. The emergent layer swings between hot days and cool nights with constant wind. The canopy stays humid and warm with little air movement. The understory remains cooler and even more humid. The floor is coolest, dampest, most stable. The forest contains multiple climate zones, stacked vertically, as distinct as different ecosystems hundreds of miles apart. This layered structure appears in forests worldwide, though specifics vary. Tropical rainforests develop the most pronounced layering, with all 4 layers clearly defined. Temperate forests show simpler stratification, often just canopy, understory, and floor. Boreal forests compress into even simpler structures, but wherever forests grow tall and dense enough, they build upward, creating vertical complexity that multiplies available habitat. A single acre of forest, if you could unfold all its surfaces, every leaf, every branch, every square inch of bark, would contain many acres worth of surface area. This 3-dimensional architecture is why forests support such extraordinary diversity. They don't just spread across the ground. They build cities in the air, stacking neighborhoods from soil to sky, each floor humming with its own distinct community of life. Picture a seedling pushing through leaf litter. Its roots probe the darkness, searching. Within days, fungal threads finer than spider silk wrap around the root tips. The fungus doesn't strangle, it merges. The 2 organisms blur their boundaries, creating what scientists call a mycorrhiza, from Greek, myco for fungus, rhiza for root. The fungus can extend its reach far beyond what the tree's roots could access alone, spreading through an area up to 1,000 times larger. These microscopic threads, called hyphae, snake between soil particles, infiltrating spaces too small for root hairs. They extract nitrogen and phosphorus from locked minerals in rock, organic compounds in decaying matter, water from distant pockets of soil. The tree offers what the fungus cannot make: sugar. Through photosynthesis, trees can transfer up to 30% of captured carbon to fungal partners. It's expensive, but necessary. Without these allies, most trees would starve. But here's where the story gets strange. That same fungus doesn't stop at one tree. Its threads continue outward, reaching toward other roots: Douglas fir, paper birch, hemlock, cedar. Some fungal networks can connect with dozens of tree species simultaneously. What emerges is not a simple partnership between 2 organisms, but a web, a forest-wide network linking hundreds of trees through millions of fungal threads woven through every cubic inch of soil. Scientists call this the mycorrhizal network, though some use a more evocative term, the wood wide web. These networks transfer information. In 1997, researcher Suzanne Simard grew Douglas fir and paper birch seedlings together, feeding one tree radioactively tagged carbon. The carbon flowed between species, from shaded birch to sun-exposed fir in summer, reversing in autumn when birch lost its leaves. Further experiments revealed stranger behavior. When researchers damaged trees or exposed them to insects, chemical signals traveled through the network to neighbors. Undamaged trees responded by increasing defensive compounds, preparing for attacks that hadn't reached them yet. Trees carry more than distress signals. Those connected to robust networks show faster growth, better drought resistance, and higher survival rates. Seedlings plugged into networks survive at dramatically higher rates, even in deep shade where minimal light reaches. Some trees act as hubs, older, larger individuals with sprawling root systems. A single hub can connect with hundreds of other trees. Simard found that removing these hubs caused cascade failures, with connected trees showing reduced growth and increased mortality. The fungal networks show something like memory. When researchers repeatedly stressed plants connected to the same mycelial network, the network's response grew more efficient over time, releasing defensive signals faster and in greater quantities. The fungus itself seemed to be learning, or at least adapting its behavior based on past events. Not all interactions are cooperative. Some fungi provide better service than others. Trees show preferences, allocating more carbon to better partners. Some fungi cheat, taking without giving. Others act as middlemen, skimming nutrients. The underground network contains cooperation, trade, exploitation, and theft. Network architecture matters. In healthy forests, the fungal web grows dense and redundant, with multiple pathways connecting every tree. Cut roads or clear patches, and the network fragments. Trees at edges show reduced growth and increased disease vulnerability. Dig into forest soil, and you'll find evidence everywhere. In a single teaspoon, fungal hyphae can stretch for dozens to hundreds of meters if laid end to end. The total length in the top inches of soil across 1 acre would extend for thousands of miles. This isn't empty space, it's dense architecture, a city built from filaments too small to see without magnification. The fungal threads aren't eternal. They die and are replaced constantly, their nutrients recycled. But the network persists through the ongoing relationship between tree and fungus, a partnership that has existed for more than 400 million years, since the first plants colonized land. Imagine a Douglas fir in British Columbia's coastal ranges, its bark scarred by beetles. Insects tunnel through its living tissue, carving egg galleries. The tree detects the invasion within hours, cells rupturing, damage signals cascading. The response begins immediately. Far below in the root system, something shifts. The tree adjusts the chemistry of sugars sent through the mycorrhizal network, encoding information into carbon molecules. Neighboring trees receive these altered compounds. Their chemistry changes. Within days, trees 50 feet away begin producing defensive compounds, bitter chemicals that make tissue less palatable, harder to digest, toxic in sufficient quantities. The beetles move to these trees and find them unexpectedly hostile. Many die. Others move on. The forest has communicated and defended itself. This isn't metaphor. Researchers have measured these changes, tracked chemical signals through soil. documented defensive responses in trees that received warnings. But the conversation goes deeper. Suzanne Simard's research revealed that hub trees don't distribute carbon evenly. They show preferences, sending more resources to certain individuals. The pattern became clear when she analyzed genetics. Hub trees sent disproportionately more carbon to their own offspring. Trees recognized their kin through chemical signals and invested more heavily in their children's survival. This changed our understanding of forest dynamics. Competition exists, but so does cooperation, especially between related individuals. A mother tree might shade out her seedlings while simultaneously subsidizing them underground with carbon, water, and nutrients. The preferences weren't absolute. Hub trees also supported unrelated seedlings, particularly during stress. When drought or disease struck, old trees increased resource transfers to struggling neighbors regardless of kinship, prioritizing forest health as a whole. When researchers severed mycorrhizal connections using trenches or fungicides, isolated individuals showed immediate stress, reduced growth, lower water efficiency, decreased disease resistance. Remove the connections, and performance declined across almost every measure. The timing matters. Carbon flows most strongly at night. Water moves constantly, pulsing in response to drought. Nitrogen transfers peak in spring when growth demands are highest. The network operates on multiple schedules simultaneously. Some trees proved more generous than others. Certain species consistently gave more than they received, acting as net donors. Others took more, though rarely so much that they damaged partners. Mycorrhizal fungi can cut off trees that drain resources without reciprocating. Hub trees occupied unique positions. Their massive root systems connected them to more fungal partners than younger trees could access, giving them broader networks and more trading partners. When these hubs died or were removed, effects rippled outward. Simard documented forests where hub removal led to increased mortality in connected seedlings years later. The trees don't just exchange resources, they synchronize. Researchers found that connected trees often massed together, producing massive seed crops in the same years, despite growing in different conditions. The coordination happens through the network, with chemical signals helping align reproductive timing across the forest. This synchronization serves a purpose. By masting together, trees overwhelm seed predators. Animals simply can't consume the entire crop, so more seeds survive. The network carries signals that help coordinate this behavior across hundreds of individuals. The conversations aren't always benevolent. Some trees send chemical signals that suppress competitor growth, releasing compounds that inhibit rival species' seed germination or slow established neighbors. Other trees eavesdrop and adjust their growth patterns, avoiding areas where chemical warfare is underway. Black walnut trees produce juglone, toxic to many plant species. They release it through roots and decomposing leaves, creating suppression zones. But juglone also travels through mycorrhizal networks, extending the tree's chemical influence. Neighboring trees connected to the same network may experience reduced growth even without direct competition. Network architecture influences who talks to whom. Trees growing close together share more fungal connections, creating neighborhoods of intense communication. But long-distance connections also exist. Fungal highways spanning dozens of yards, linking distant individuals and providing redundancy if local connections sever. Age matters. Young trees tend to be net receivers, pulling resources while contributing little. As they mature, the balance shifts. They contribute more than they take, eventually becoming net donors. The oldest trees give the most, transferring massive amounts to younger generations. This creates generational flow, with elders supporting youngsters who will eventually support the next generation. But the system isn't infinitely generous. During extreme stress, severe drought, widespread disease, resource scarcity, the network contracts. Trees reduce donations, prioritizing their own survival. The most stressed individuals may be abandoned, cut off as the network reallocates resources to healthier members. It's triage, sacrificing the vulnerable to preserve community health. These decisions aren't conscious. Trees don't deliberate, but the chemical and physiological mechanisms governing resource allocation create behavior that looks strategic, even cooperative. The tree sharing carbon isn't being altruistic. It's following biochemical rules shaped by millions of years of evolution. But the effect is the same. Forests function as communities, not just collections of individuals, because their members are literally connected, constantly exchanging resources and information through hidden networks beneath the soil. Stand in a forest, and you're standing amid countless conversations, chemical phrases passing through fungal threads at your feet, trees sharing news, coordinating growth, supporting young, warning neighbors, remembering kin. The forest talks. We're only beginning to learn its language. Kneel on the forest floor and brush aside the top leaves. Beneath lies another layer, partially broken down, its structure softening. Below that, leaves have lost their shape, fragmenting into pieces. Deeper still, the material becomes dark, crumbly, smelling of earth. You're watching matter move backward through its history, complexity dissolving into simplicity. Except it's not chaos. It's transformation, orchestrated by the most diverse workforce on the planet. A single teaspoon of forest soil contains more individual organisms than people on Earth. Bacteria and fungi work alongside each other, each species filling specific roles in deconstruction. These microorganisms operate in sequence, each preparing the ground for the next wave. When a leaf falls, it carries the chemical signature of the tree that made it. Fresh leaves contain simple sugars, amino acids, and starches, easily digestible compounds that attract fast-growing bacteria and fungi. These opportunists arrive within hours, multiplying rapidly, consuming the available nutrients. The fungi secrete enzymes, molecular scissors snipping apart chemical bonds, holding the leaf together. Different enzymes target different compounds. Cellulases break down cellulose, the structural material giving plant cells rigidity. Ligninases attack lignin, the polymer making wood woody. Proteases dismantle proteins, releasing nitrogen. As early colonizers work, they soften the leaf, making it accessible to organisms with less sophisticated toolkits. Small arthropods, springtails, mites, millipedes move in, chewing the partly decomposed tissue. They don't digest most of what they eat. Instead, they shred material into smaller pieces, dramatically increasing surface area. A leaf fragment passing through a millipede emerges in dozens of tiny pellets, each exposing fresh surfaces for microbes. The microbial community shifts as easily digested compounds disappear. Fast-growing generalists give way to specialists equipped with enzymes for tougher materials. Bacteria with plant cell wall-degrading genes work more slowly than initial colonizers, but they access nutrients locked in structures the first wave couldn't touch. Fungi become increasingly important as decomposition progresses. Their filamentous structure allows them to bridge gaps between nutrient patches, transporting resources from one location to another. Some fungi specialize in lignin degradation, a process few organisms can accomplish. Lignin's molecular structure resists most attempts to break it down, but certain fungi produce enzymes that oxidize its bonds. Wood decomposition proceeds even more slowly. A fallen trunk might take decades to disappear. The process follows a predictable sequence. Bark beetles and wood-boring insects tunnel through outer layers, creating galleries that expose inner wood. Fungi follow, their spores germinating in insect tunnels, hyphae spreading through wood grain. Different fungi colonize wood at different stages. Some attack cellulose first, leaving lignin behind. Others specialize in lignin, creating spongy, bleached wood that crumbles easily. The type of fungi determines the wood's appearance as it decays. In coniferous forests, fungi quantitatively dominate the microbial community in the litter horizon. This stratification reflects changing chemistry of decomposing material. Fresh litter contains complex polymers fungi excel at breaking down. Deeper in soil, where decomposition has extracted these compounds, bacteria become relatively more important. Not all decomposers are microscopic. Earthworms play an outsized role in some forests, pulling leaf litter into burrows, mixing organic matter with mineral soil, processing both through their digestive systems. Their castings contain higher nutrient concentrations than surrounding soil. In forests where earthworms thrive, they can process the entire leaf litter layer in a single season. But earthworms aren't universal. Many forests evolved without them, particularly in regions glaciated during ice ages. In these places, decomposition proceeds more slowly. Organic matter accumulates in thick layers, and soil structure differs dramatically. The microbial communities work in concert with the living forest. Many fungi both form partnerships with tree roots and break down dead organic matter, simultaneously destroying old plant tissue while feeding living plants. Bacteria decompose dead fungal threads, creating feedback loops where nutrients cycle continuously through living and dead tissues. Temperature and moisture profoundly affect decomposition rates. In tropical rainforests, where heat and humidity stay high year-round, decomposition proceeds rapidly. A fallen leaf might vanish in weeks. In boreal forests, where winters freeze the soil solid for months, decomposition slows dramatically. The same leaf might persist for years. This difference in decomposition rate explains why tropical soils are often nutrient-poor, despite the luxuriant growth above them. Nutrients cycle so quickly that little accumulates in the soil. Boreal soils, conversely, build thick organic layers because decomposition can't keep pace with leaf fall. Seasonality introduces rhythm to the decomposition process. In winter periods without photosynthate flow from plants, decomposer fungal species show increased activity. When trees photosynthesize actively during the growing season, they send sugars underground to their mycorrhizal partners, supporting that network. When photosynthesis stops in winter, the mycorrhizal fungi reduce activity, and decomposer fungi, fed by dead organic matter, rather than living roots, take center stage. The chemistry of what's being decomposed matters enormously. Different tree species produce leaves with vastly different compositions. Oak leaves contain high levels of lignin and tannins, making them slow to decompose. Aspen leaves break down much faster, with less lignin and more nitrogen. Conifer needles fall somewhere in between, but contain acidic compounds that further slow decomposition. These differences mean that the forest floor beneath an oak grove looks and functions differently from the floor beneath aspens, even if both forests grow in similar climates. The ratio of carbon to nitrogen in organic matter, the CN ratio, determines how quickly decomposers can process it. Decomposers need nitrogen to build their own bodies, but plant tissue often contains much more carbon than nitrogen. When microbes encounter high CN material, they must pull nitrogen from the soil to supplement what the plant material provides. This can temporarily lock up nitrogen, making it unavailable to plants, a phenomenon called nitrogen immobilization. As decomposition proceeds and the C:(N) ratio drops, microbes eventually release excess nitrogen back to the soil, where plant roots can absorb it. This creates an interesting pattern in nutrient availability over time. Immediately after a large input of high CN material, say, a major windstorm that knocks down many trees, nitrogen availability drops as decomposers tie it up in their biomass. Years later, as those decomposers die and are themselves decomposed, nitrogen floods back into the system. The forest experiences pulses of scarcity and abundance, driven by the dynamics of decomposition. Human activities have altered decomposition in ways we're only beginning to understand. Nitrogen pollution from vehicle exhaust and agriculture has increased nitrogen deposition in many forests, changing the CN ratios of leaf litter and speeding decomposition. This sounds beneficial. Faster nutrient cycling should support more growth, but it also means less carbon storage in soil, as organic matter breaks down before it can form stable soil compounds. The carbon that would have stayed locked in soil for decades or centuries now returns to the atmosphere much faster. Climate change is shifting decomposition patterns more fundamentally. Warmer temperatures accelerate microbial activity, speeding decomposition, but different organisms respond differently to warming. Bacteria tend to increase activity more than fungi with rising temperatures. This could shift the balance between these groups, changing which compounds get decomposed, in which order, potentially altering soil structure and nutrient cycling in ways that cascade through the entire forest ecosystem. Soil microbial communities respond to forest degradation by forming more complex interaction networks, possibly as an adaptation to resist nutrient loss. When forests are stressed or damaged, the decomposer community reorganizes, creating more connections between species, more redundancy in who can perform critical functions. This flexibility suggests resilience, the capacity to maintain function even as conditions shift. But resilience has limits. Severe disturbances can overwhelm the decomposer community's ability to adapt. Clear cutting exposes soil to direct sunlight, drying it out and heating it up, killing many decomposers outright. Those that survive face altered conditions, different moisture, different temperature, different inputs of organic matter. The community that eventually establishes may function differently from the original, cycling nutrients on different timescales, storing carbon less efficiently, supporting different plant communities. Stand in a forest and you stand on a graveyard being continuously recycled. Every step you take presses down on the remains of countless organisms, trees that fell decades ago, leaves that dropped last autumn, insects that died yesterday, all being systematically disassembled by organisms too small to see without magnification. This invisible workforce, operating in darkness, in silence, in numbers beyond comprehension, performs the essential work of closure, transforming the dead into the building blocks of new life, ensuring that nothing is truly final, that every ending feeds a beginning. The forest waits in darkness. No wind moves through the canopy. The temperature has dropped to the coldest hour just before sunrise. The air hangs still and heavy with moisture. Then, from somewhere in the understory, a single voice breaks the silence. A robin begins to sing. The sound carries through the dark forest, moving farther than it would at any other time of day. Within minutes, other robins answer from distant territories. More voices join, blackbirds, thrushes, each species entering at its appointed time, as if following a score written into their biology. This is the dawn chorus, and it happens every morning in forests around the world. The timing is precise. Each species begins singing at a specific time before sunrise, some 45 minutes early, others 30, still others waiting until first light touches the canopy. The sequence rarely varies. In European forests, robins and blackbirds start first, followed by wood pigeons and wrens, with finches and sparrows joining only when enough light has arrived. Why dawn? The obvious answer seems acoustic. Sound carries roughly 20 times farther in the early morning than later in the day. Cool air creates conditions where sound waves bend downward rather than dispersing upward. Humidity is higher before sunrise, and moisture transmits sound more efficiently. Wind hasn't yet begun to stir. But recent research suggests the acoustic advantage alone doesn't fully explain the chorus. When scientists analyzed tropical rainforest recordings comparing dawn and dusk, they found something unexpected. If birds sang primarily because morning air carries sound better, then species using higher frequency songs should show the strongest preference for dawn over dusk. Researchers found no significant relationship between song frequency and dawn preference. The acoustic hypothesis, while intuitively appealing, couldn't account for observed patterns. So if not just acoustics, then what? Multiple factors converge at dawn. The light level matters enormously. It's too dark to forage efficiently. Most insects aren't active yet, and birds can't see well enough to hunt, but it's also too dark for most predators. Hawks and other raptors hunt by sight and need good light. The pre-dawn twilight offers birds a relatively safe period when they can expose themselves through song without as much risk. Energy considerations also play a role. Singing is expensive. A bird in full song can increase its metabolic rate substantially. But birds need to sing. It's how they defend territories and attract mates. Dawn offers a solution. After a night of fasting, birds have empty stomachs and depleted energy reserves. They can't forage yet because it's too dark. So they allocate remaining energy to singing, making the most of this otherwise unproductive time. Only the strongest, healthiest males can produce the loudest songs on an empty stomach after a cold night, so song quality becomes an honest signal of fitness. The hormonal dimension, adds another layer. As day length increases in spring, it triggers brain changes in birds. Longer days trigger hormonal changes that enlarge parts of the brain controlling song production. Melatonin levels drop as light increases, initiating a cascade of hormonal activity that primes birds for vocal behavior. This happens most strongly at dawn, when the shift from darkness to light is most rapid. Territory maintenance drives much of the singing. Male birds establish territories during breeding season, patches of forest they defend against rivals. These territories contain resources needed for nesting and raising young, food, suitable nest sites, cover from predators. A male who can hold a good territory has a much better chance of attracting a mate, but territories require constant assertion, constant reminder to neighbors that this space is occupied and defended. Dawn singing serves this perfectly. By singing at first light, a male announces to neighbors that he survived the night. He's still strong enough to sing vigorously, and he still claims this territory. It's a daily roll call. Rivals listening from adjacent territories receive this information and adjust accordingly. A male who fails to sing one morning signals weakness, and neighbors may probe into his territory, testing whether they can claim it. Mate attraction provides the other major motivation. Female birds evaluate potential mates partly through song quality. A male with a complex, loud, sustained song demonstrates he has good genes for producing the neural and physical machinery of song. He's healthy enough to maintain that machinery, and he holds a territory with enough resources to support the energy demands of singing. Females listen and compare, often choosing males with the most impressive displays. The timing of individual species entries into the chorus isn't random. Robins, skylarks, song thrushes, and blackbirds are among the earliest singers, while warblers and wrens join later. This staggered timing may help species avoid acoustic interference. If every species sang simultaneously, individual songs would blur into noise. By singing at different times, species create acoustic niches, temporal slots where their particular frequency and rhythm stand out. Within species, males also time their songs to avoid overlap. Researchers have found that neighboring males of the same species often synchronize their singing in a specific pattern. They take turns. One male sings, waits for a response, sings again, only after the neighbor has replied. This turn-taking allows each bird's song to be heard clearly without competition from immediate neighbors. Some species produce songs heard only during the dawn chorus, special vocalizations they don't use at other times. These dawn-specific songs tend to be longer, more complex, and louder. The investment makes sense, given the importance of the moment. Dawn is when the most rivals are listening, when acoustic conditions are optimal, when the most females are evaluating potential mates. The intensity varies with season. In temperate forests, the chorus peaks during spring breeding season, particularly in May and June, when most species are establishing territories and attracting mates. As summer progresses and breeding winds down, the chorus quiets. By late summer, many species have stopped singing entirely, their territorial and mating needs met. Weather affects the chorus as well. On still, clear mornings, singing reaches maximum intensity. Storms suppress it. Birds conserve energy during bad weather, and rain creates acoustic interference. Temperature matters too. Very cold mornings reduce participation, especially among smaller birds who lose body heat rapidly. The chorus isn't purely cooperative. Competition exists within it. Males sing louder to be heard over neighbors. Some species have evolved strategies for acoustic dominance, producing songs at frequencies or rhythms that make them stand out. Others use volume, simply outshouting rivals. Still others rely on complexity, producing elaborate songs that demonstrate superior neural capacity. Urban environments are changing the dawn chorus in measurable ways. Artificial light advances dawn singing by 10 to 21 minutes in species like robins and blackbirds. Birds near streetlights begin singing earlier than those in darker areas, responding to artificial light as if dawn has arrived. This shift can create mismatches, birds singing when acoustic conditions aren't yet optimal, or when predators are still active. Noise pollution also affects the chorus. In forests near roads or urban areas, birds adjust their songs to compete with ambient noise. Some sing louder, others shift to higher frequencies that cut through low-frequency traffic noise. These adjustments require energy and may reduce song quality in other dimensions. The dawn chorus exists in counterpoint to a dusk chorus, birds singing at sunset. But the evening performance is quieter, less intense. The same acoustic advantages don't apply. Evening air is warmer, windier, drier, and the motivations differ. By evening, birds have spent the day foraging and have full stomachs, but predators are still active at dusk, making extended vocal displays riskier. The New Zealand forest once had a particularly loud dawn chorus, but extensive forest loss and introduced predators have reduced its intensity. This pattern repeats globally. Wherever forests are fragmented or degraded, the dawn chorus diminishes. Fewer birds mean fewer voices. Simplified forest structure means poorer acoustic conditions. Stand in a healthy forest 30 minutes before sunrise on a May morning, and you witness one of the planet's most ancient performances. The songs you hear are refined by millions of years of evolution, each trill and warble shaped by countless generations of competition and selection. The timing, the sequence, the intensity, all finely tuned to exploit a brief window when acoustic conditions, safety, and biological drive align perfectly. The forest is never truly silent, but dawn is when it speaks most clearly. The chorus is conversation and competition, cooperation and conflict, all woven into a layered acoustic fabric that announces the forest's health, marks the boundaries of invisible territories, and carries the messages that will determine which birds breed and which genetic lines continue. Its commerce conducted in song, real estate claims broadcast in melody, courtship carried out through rhythm and tone. Listen closely, and you're hearing the forest negotiate with itself, working out through sound who will live where, who will mate with whom, which individuals and which species will claim resources and pass their genes forward. The dawn chorus is the forest thinking out loud. A caterpillar chews through an oak leaf, its mandibles slicing cells open. The damage triggers an immediate cascade inside the leaf. Wounded cells release enzymes, breaking down fatty acids in cell membranes. Within minutes, the leaf starts producing a cocktail of volatile organic compounds, molecules light enough to evaporate at normal temperatures. These aren't random chemicals. The leaf manufactures specific compounds in response to specific damage types. A tear from wind produces one signature. A fungal infection produces another. Herbivore damage. Saliva mixing with torn tissue produces yet another distinct chemical profile. The oak is sending a message specifying exactly what threat it faces. The volatiles drift on air currents, spreading through the forest. Some compounds travel only a few meters before breaking down. Others persist longer, carrying farther. Within an hour, trees 50 feet away begin responding to chemicals they didn't produce, preparing defenses against attacks that haven't reached them yet. Plants respond to stimuli by emitting volatile organic compounds that provide information on the emitter plant's physiological status. This isn't metaphor. Receiving plants detect these airborne chemicals through leaf surfaces and respond with measurable physiological changes. The chemistry involved is complex, but follows recognizable patterns. When insects chew leaves, plants rapidly produce green leaf volatiles, 6-carbon alcohols and aldehydes that smell fresh and grassy. These compounds form from immediate breakdown of damaged membranes, representing the plant's fastest response to injury. Following quickly come the terpenes, molecules built from 5 carbon units that can combine in countless ways. 2 monoterpenes and 2 sesquiterpenes, known to have cytotoxic and deterrent effects, were the most frequent compounds found in tropical tree surveys. These chemicals serve double duty. Some directly repel insects or inhibit digestion, while others act as signals to neighbors or beacons, attracting insect predators. The receiving plants don't just passively detect these signals. Many green leaf volatiles promote jasmonic acid levels in receiver plants, which prepare defenses for subsequent herbivore attack. Jasmonic acid is a plant hormone activating defensive genes throughout the plant, triggering production of compounds that make leaves tougher, more bitter, or outright toxic to insects. The sophistication becomes clear when you examine timing. Trees don't maintain maximum defenses constantly. That would waste too much energy. Instead, they operate in a state of readiness, able to ramp up defenses quickly when threatened. Volatile signals from damaged neighbors allow them to activate defenses before being attacked themselves, gaining precious time. This phenomenon is called priming. Volatile organic compounds enable plants to communicate across distances. And when a plant is damaged by pests, these compounds can be detected by neighbors, prompting them to enhance defenses. A primed plant hasn't invested heavily in defenses yet, but it has readied its molecular machinery to respond faster and stronger when attack comes. The chemical conversations extend beyond simple danger warnings. Some volatiles attract predators and parasites of herbivorous insects. When a caterpillar feeds on a tree, the tree releases compounds that summon wasps, species that lay eggs inside caterpillars. The wasp larvae consume the caterpillar from within. The tree effectively calls for reinforcements, using chemistry to recruit allies. The biochemical composition of forest air tends to follow diurnal variations, with volatile organic compound concentrations usually peaking early afternoon and sometimes in morning around 2 hours after dawn. This daily rhythm reflects tree metabolic cycles. Photosynthesis increases during morning, providing energy for volatile production, while temperature rises through midday, increasing evaporation rates. Not all chemical signals travel through air. Several studies have shown important roles for passively mediated interactions involving adsorption of chemicals to plant surfaces. Heavier molecules, particularly sesquiterpenes, don't evaporate completely. Instead, they settle onto nearby leaves, coating them with chemical film. Plants receiving this coating can gain enhanced resistance to herbivores. The diversity of these compounds staggers the imagination. A survey discovered remarkably high chemical diversity, with 264 distinct volatile organic compounds and a mean of 37 compounds per species among tropical rainforest trees. Each species has its own chemical vocabulary, and some can adjust emissions based on what attacked them. Trees even show chemical recognition of kinship. Research has found some species increase volatile emissions when growing near relatives, possibly amplifying warning signals to family members. Recognition mechanisms likely involve chemical detection through roots or mycorrhizal networks. The chemical conversations aren't always benign. Some trees release volatiles that suppress competitor growth, creating zones of chemical dominance. Black walnut produces juglone, which inhibits many other plant species. Eucalyptus trees release terpenes that accumulate in soil and prevent other species' seeds from germinating. These chemical weapons shape forest composition. Temperature affects how chemical conversations unfold. Warmer conditions increase production and evaporation of volatiles, potentially accelerating signal spread. But heat also speeds breakdown. Atmospheric oxidants react with volatiles faster at higher temperatures, potentially reducing signal travel distance. Climate change may be altering effective range of plant communication. Air pollution interferes with these signals in concerning ways. Ozone and other pollutants react with plant volatiles, breaking them down before they reach neighbors. In polluted areas, chemical warnings may travel shorter distances or arrive degraded. This could leave forests more vulnerable to herbivore outbreaks. Once emitted, volatiles are absorbed through stomata and diffuse across mesophyll cells of neighboring plants, with response involving intricate intracellular and intercellular signaling mechanisms, including calcium fluxes. The detection system is elegant. Specialized receptors on cell membranes bind to specific volatile molecules, triggering cascades of internal signals that ultimately reach the nucleus and activate defensive genes. Some of these genes encode enzymes that produce more volatiles, creating a positive feedback loop. A plant receiving a danger signal begins emitting its own warnings, amplifying the message and spreading it farther. This can create waves of defensive preparation moving outward from initial attack sites, priming trees across large areas. The time scales matter. Green leaf volatiles release immediately, within seconds to minutes. Terpene production takes longer, ramping up over hours. As a result of attack, the induced production of volatiles is localized to the damage site, but systemic induction has also been detected. The plant activates defenses both at the wound and in distant, undamaged tissues. The volatiles don't just defend against herbivores. Some protect against fungal pathogens through direct antimicrobial effects or by priming antifungal defenses in neighbors. The chemical conversations serve multiple functions simultaneously: warning, coordinating, defending, competing, creating a complex informational landscape overlaying the physical forest. Human uses of forest volatiles extend back millennia. Essential oils extracted from pine, eucalyptus, cedar, and countless other trees contain the same defensive compounds trees use to protect themselves. We harness their antimicrobial properties in medicines, their repellent effects in insect deterrence, their aromatic qualities in perfumes. Modern research explores whether exposure to forest volatiles provides health benefits, reduced stress, improved immune function, enhanced cognitive performance. Whether forests evolved these chemicals solely for defense or whether they serve additional ecological functions remains debated. What's clear is that forest air is thick with messages, a continuous exchange of molecular information flowing between trees, shaping behavior, coordinating defenses, mediating competition. We walk through forests breathing this chemical conversation, usually unaware that we're inhaling the vocabulary of an ancient language spoken in volatile compounds, a dialogue ongoing since the first forests covered the first continents. Stand in a forest and close your eyes. That scent, pine resin, damp earth, decaying leaves, is more than pleasant aroma. It's active communication, trees broadcasting their status, warning neighbors, summoning allies, marking territory, coordinating responses to threats. The forest is talking in chemistry, and though we can't decode most of what's being said, the conversation never stops. The air never stops carrying these molecular messages, information flowing through the unseen chemical dimension of forest ecology. A single large oak tree stands in a temperate forest, its canopy spreading 40 feet across. During the growing season, this tree can pull up to 100 gallons of water from the soil through its roots on a hot summer day. The water travels upward through channels in the trunk, xylem vessels that function like microscopic pipes, stacked end to end, creating continuous pathways from root tip to leaf edge. The journey can span 100 feet or more in tall trees, water climbing against gravity through a combination of forces so finely balanced that breaking a single column of water in the xylem creates tension throughout the entire system. The water doesn't stay in leaves. Most evaporates through tiny pores called stomata, openings scattered across the leaf surface, each surrounded by guard cells that can open or close the pore in response to light, humidity, and the tree's internal water status. When stomata open to allow carbon dioxide in for photosynthesis, water vapor escapes. This process, transpiration, is essentially unavoidable. The tree needs carbon dioxide to build sugars, but accessing it means exposing moist internal tissues to dry air. Water follows the concentration gradient, evaporating continuously as long as stomata remain open. A large oak tree can transpire 40,000 gallons per year, which averages to over 100 gallons per day during the growing season. European forests transpire about 400 millimeters per year, or roughly half of the average rainfall. This isn't water wasted. It's water being actively cycled back into the atmosphere, where it will eventually condense and fall as rain, possibly on the same forest, possibly hundreds of miles away. The cooling effect of this evaporation is substantial. An individual tree transpiring 100 gallons on a hot day provides cooling equivalent to running an air conditioner for several hours. Multiply that by millions of trees, and you begin to understand why forests maintain cooler, more stable temperatures than cleared land. The energy required to evaporate water comes from the surrounding environment, heat absorbed from the air, from sunlight striking leaves, from the tree's own metabolic processes. This energy leaves the system as latent heat, embedded in water vapor, only releasing when that vapor eventually condenses back into liquid water, possibly miles away. Forests don't just participate in the water cycle, they drive it. On a typical day, the trees in the Amazon release 20 billion tons of moisture into the atmosphere. This moisture doesn't dissipate randomly. It forms patterns, flows, invisible rivers carrying more water than some terrestrial rivers. The concept of moisture recycling challenges our usual picture of the water cycle. We imagine rain coming from the ocean. Water evaporates from the sea, forms clouds, blows inland, falls as rain. That's accurate for coastal regions. But deep in continents, far from oceans, a different pattern emerges. The interiors of continents often get most of their precipitation from water that has been rained out and recycled back into the air several times in a precipitation cascade following the winds. The farther inland you go, the more dominant this recycling becomes. Trees are the engines of this recycling system. Their roots reach deep into soil layers that surface evaporation can't access. They pull water from depths of 10, 20, sometimes 30 feet, water that would otherwise remain locked underground. By transpiring this deep water into the atmosphere, trees effectively short-circuit the usual water cycle, bypassing the slow process of groundwater flow and surface evaporation, injecting moisture directly into the air, where it's immediately available to form clouds. Ari Stahl of Wageningen University reported that 1/3 of the rain falling in the Amazon basin comes from moisture generated within the basin, mostly by transpiring trees. The dependence grows stronger as you move inland, away from the Atlantic Ocean. Western regions of the Amazon receive most of their rain from moisture that has already fallen and been transpired back into the atmosphere multiple times. Remove the trees, and you remove the mechanism that keeps rain falling in the interior. This creates what researchers call the biotic pump. Regions with higher leaf area are expected to attract winds from areas with lower leaf area, because transpiring vegetation creates zones of rising air and lower pressure. Air flows toward these low-pressure zones, bringing moisture with it. The arriving moisture condenses as it rises over the forest, falling as rain, getting absorbed by trees, and transpiring back into the atmosphere to continue the cycle. The evidence for this appears in rainfall patterns downwind of forests. In a study of tropical areas downwind of deforestation, researchers found that air that has passed over extensive vegetation in the preceding few days produces at least twice as much rain as air that has passed over little vegetation. The effect extends remarkable distances. Studies have found that the Amazon's moisture cycle regulates rainfall patterns as far as the Midwest in the United States. Forest loss disrupts these patterns in predictable ways. Sao Paulo, the largest city in Brazil, and just a 4-hour plane ride from the Amazon rainforest, has recently been suffering from one of the worst droughts in the country's recent history, a direct effect of deforestation in the Amazon. The mechanism is clear. Fewer trees means less transpiration, less atmospheric moisture, reduced rainfall downwind. The effects cascade. Reduced rainfall means drier conditions, increased fire risk, more tree death, further reduction in transpiration, leading to even less rain. The timing of transpiration adds complexity to this picture. Stomata don't stay open continuously. Many tropical trees close their stomata after a few hours of intense photosynthesis, responding to temporary depletion of available soil water. This creates daily rhythms in moisture release. Some research suggests cyclic patterns of transpiration with periods of roughly 70 to 80 minutes, creating pulses of moisture release rather than a steady flow. These rhythms may influence cloud formation timing, though the mechanisms remain unclear. Trees also transpire at night, though at much lower rates than during the day. Stomata partially close in darkness, but they don't seal completely. Water continues escaping, driven by the moisture gradient between leaf interior and outside air. This nocturnal transpiration varies by species and environmental conditions, and its ecological function remains debated. It may help maintain nutrient flow through the xylem or prevent excessive buildup of carbon dioxide inside leaves, or simply be an unavoidable consequence of maintaining partially open stomata for rapid morning response when photosynthesis resumes. The forest canopy intercepts rain before it reaches the ground. Leaves, branches, bark, all these surfaces catch water, holding it temporarily. Some evaporates directly back to the atmosphere without ever touching soil. The rest drips gradually to the forest floor or runs down tree trunks. This interception smooths the impact of rainfall, instead of rain striking the ground with full force, potentially causing erosion. It arrives gently, its energy dissipated by passage through the canopy. Tree and forest root systems contribute to better soil water infiltration, and their large leaf litter production leads to more soil carbon, which improves both water retention in the soil and groundwater recharge. Forest soils function as sponges, absorbing rain and releasing it slowly to streams over days or weeks, rather than hours. This buffering effect means forested watersheds maintain more stable stream flows year-round compared to cleared areas, which experience flash flooding during storms and low flows during dry periods. The particles that trees release into the atmosphere, fungal spores, pollen, bacteria, organic compounds, serve another crucial function. Water vapor needs something to condense onto. In clean air, condensation requires very high humidity levels. But when tiny particles are present, moisture condenses more easily at lower humidity thresholds. Trees effectively seed the atmosphere with condensation nuclei, making it easier for their transpired moisture to form clouds. Forest structure affects these processes in measurable ways. Dense, closed canopy forests with multiple layers transpire differently than open forests with scattered trees. Research comparing disturbed and undisturbed forests shows that canopy gaps create warmer, drier conditions with increased vertical mixing of air. The arrangement of trees influences how light and heat reach the ground, affecting not just transpiration, but also evaporation from soil and leaf litter. Climate change is altering forest water cycling in multiple directions simultaneously. Rising carbon dioxide concentrations affect stomatal behavior. Many plants partially close stomata in response to elevated carbon dioxide, reducing water loss. This sounds beneficial, but it also means reduced transpiration, less moisture returning to the atmosphere, potentially less rainfall downwind. Warmer temperatures increase evaporation rates and transpiration potential, but they also increase atmospheric vapor pressure deficits, making it harder for plants to keep stomata open without suffering water stress. The feedback loops become concerning. Reduced rainfall stresses forests, causing trees to close stomata or shed leaves to conserve water. This reduces transpiration, leading to even less rainfall. The system can spiral into self-reinforcing drought, potentially flipping forests into alternative states: savanna, scrubland, eventually desert. Some models suggest that continued deforestation in the Amazon could push the system past a tipping point where the forest can no longer generate enough rainfall to sustain itself, leading to widespread dieback, even in currently protected areas. Water quality flows from forest cover. As rain filters through forest soil, microorganisms break down pollutants, tree roots absorb excess nutrients, and the soil matrix physically filters sediments. Many cities source drinking water from forested watersheds, specifically because the natural filtration is so effective. New York City draws most of its water from the Catskill Mountains watershed, avoiding billions in treatment costs by maintaining forest cover that naturally purifies the water. The ancient understanding of forests as rainmakers, dismissed by early scientific forestry as superstition, turns out to contain truth. Forests don't just respond to rainfall, they create conditions that generate rainfall. The relationship is recursive, self-reinforcing. Forests need rain to grow, but they also produce rain through transpiration. Remove the forest, and you remove a critical component of the regional water cycle, potentially transitioning the area toward drier equilibrium states. Stand in a forest after rain and watch the mist rising from leaves as the sun breaks through. That visible moisture, water vapor condensing in humid air, is the forest breathing out, returning to the sky the rain that fell hours earlier. The forest is a biological pump, moving water from deep soil to high atmosphere, connecting underground reservoirs to overhead clouds, ensuring that rain falls not just once, but repeatedly, recycling through the system, sustained by the continuous hydraulic work of millions of trees, pulling water upward and releasing it outward, maintaining the moisture that makes forests possible. A chemical signal travels through the mycorrhizal network at roughly 1 centimeter per hour. An electrical impulse moves through tree tissues at about 1 centimeter per second. A bird's song crosses the forest in fractions of a second. Water moves from root to leaf in hours or days. A leaf lives for months. A tree might live for centuries. The forest operates on all these timescales simultaneously, fast events nested within slow ones, creating temporal complexity, matching the spatial complexity of the layered canopy. Stand in one spot and watch the forest change. In seconds, you see leaves trembling in wind, insects flying, birds hopping. In minutes, you notice clouds moving, shadows shifting. In hours, temperature drops, humidity rises, the forest transitions from day to night. In days, flowers open and close, fruits ripen, fungi sprout. In weeks, leaves unfold in spring or change color in autumn. In months, entire seasons pass. But you'd need years to watch slower rhythms. Trees grow imperceptibly on daily timescales. A trunk adds perhaps half an inch of diameter per year, growth you couldn't detect watching for weeks. Even the fastest trees take years to double in height, decades to reach the canopy. Death operates on similar timescales. A healthy tree might live 50 years, 100, 200. A Douglas fir can persist for 700 years. Bristlecone pines survive for millennia, their twisted trunks recording thousands of years of climate history in growth rings. These individuals aren't just old, they're archives, containing information about conditions existing before written history. The forest as a whole cycles through even longer patterns. After a disturbance clears an area, fire, windstorm, logging, succession begins. Grasses and herbaceous plants dominate for the first year or two, followed by larger species and shrubs around the third year, with tree seedlings becoming established within less than 10 years. The timeline varies by location and climate, but the sequence remains consistent. Early tree colonizers like Virginia pine, eastern red cedar, and persimmon typically appear first on dry sites, while moist sites favor loblolly, pine, sweet gum, yellow poplar, red maple, and winged elm. These pioneers grow quickly in highlight, their strategy built around speed, germinate fast, grow tall quickly, produce seeds early. The pioneer forest changes the environment beneath it. The canopy blocks light. Trees extract water from deep soil. Roots alter soil chemistry. Fallen needles and leaves acidify surfaces. These changes favor different species, shade-tolerant trees that can germinate and grow slowly under the pioneer canopy, waiting for gaps. Forest ecologists recognize 4 phases of succession: stand initiation, stem exclusion, understory reinitiation, and steady state. The first phase sees rapid establishment of vegetation. Competition intensifies during stem exclusion as trees grow taller and denser, crowding out weaker individuals. This phase typically lasts 20 to 30 years. Understory reinitiation begins when some pioneer trees die, creating small gaps in the canopy. Light penetrates these openings, allowing shade-tolerant species to establish beneath remaining pioneers. A 2-layered forest develops, fast-growing pioneers forming the upper canopy, slower-growing but longer-lived species filling the understory and midstory. Over decades, the pioneers age and die, their places taken by understory trees that have been waiting patiently. The steady state phase represents the forest's mature condition, a mix of ages and species, gaps constantly opening and closing as old trees die and young ones replace them. This isn't static equilibrium. Individual trees grow, compete, die, and are replaced continuously, but overall structure and composition remain relatively stable. How long this takes depends on location. In the North Carolina Piedmont, disturbed land can grow back to mixed hardwood forest in a century or 2. Tropical forests may reach mature structure faster, their rapid growth driven by year-round warmth and moisture. Boreal forests move more slowly, constrained by short growing seasons. But everywhere, the timescale is measured in decades to centuries, far longer than human attention spans. This creates challenges for management and conservation. We make decisions about forests on timescales of years or decades, timber harvest rotations, management plans, political terms. But forests respond on timescales of centuries. Cut down a mature forest and replant immediately, but the trees won't reach comparable size for 50 to 100 years. The complex structure of the original forest takes even longer to redevelop. A climax community persists until disturbance occurs, but in many ecosystems, disturbance happens frequently enough that multiple community types exist across the landscape. Fire-prone regions maintain mosaics of different successional stages, recent burns covered in grass and shrubs, young forests of pioneer species, patches of mature forest. This heterogeneity supports greater diversity than uniform mature forest would. Some animals specialize on early succession habitats. The American woodcock needs young forest with dense shrub cover. Golden-winged warblers require scrubby vegetation. These species decline when forests mature uniformly. Others depend on old forest. The red cockaded woodpecker requires mature longleaf pine trees at least 60 years old, and loss of old growth stands significantly threatens this species. Disturbance resets succession, but the type matters. Fire releases minerals stored in wood, creating nutrient-rich soil, while leaving standing dead trees for wildlife habitat. Logging removes wood along with nutrients. Wind throws trees over, creating tip-up mounds. Insect outbreaks kill trees, but leave structure standing. Secondary succession occurs where soil and organic matter exist, such as after fire or logging, beginning with fast-growing early successional species. This differs from primary succession on bare rock, where organisms must start from scratch, often taking centuries just to build enough soil for trees. The memory of past disturbances persists in forest structure. Walk through older forest, and you can read history in trees around you. A cohort of similar-aged oaks suggests a fire that cleared understory decades ago. A patch of white pines indicates old field abandonment. Multi-aged stands reveal forests experiencing frequent small disturbances, creating continuous regeneration. Tree rings encode climate history with annual precision. Wide rings indicate good growing years, warm, moist conditions. Narrow rings mark drought, cold, or defoliation. Fire scars appear as charred marks in wood, dating past burns. By cross-dating trees, matching ring patterns across multiple individuals, researchers construct climate records extending back thousands of years. The forest floor tells slower stories. Deep organic layers indicate centuries without major fire. Charcoal fragments reveal ancient burns. Pollen composition preserved in soil shows which species dominated in the past, allowing reconstruction of forest composition from before human records. Human-accelerated change now disrupts these ancient rhythms. Climate is shifting faster than at any point in the last 10,000 years. Trees adapted to conditions that persisted for millennia now face environments moving outside the ranges they evolved to tolerate. Some species might migrate, tracking their preferred climate as it shifts poleward and upward. But migration requires time. Trees must produce seeds. Those seeds must disperse. Seedlings must establish and grow to maturity, all while conditions continue changing. The math doesn't favor slow-growing species. A tree species might need to shift its range several hundred miles north to track optimal climate. If seeds disperse only a few dozen yards per generation, and generations span decades, migration proceeds at perhaps a mile per century. Meanwhile, climate zones shift at rates of several miles per decade. The trees can't keep up. What happens to forests outpaced by climate change remains uncertain. Some trees might adapt in place through evolution. Others might persist in microclimates, north-facing slopes, valley bottoms where local conditions buffer against regional change. Many will likely decline, replaced by species better suited to new conditions. The temporal mismatch extends beyond climate. Forests evolved with disturbance regimes, operating on natural frequencies, fire returning every few decades or centuries, pest outbreaks, cycling through populations. Human activities have altered these frequencies. Fire suppression has lengthened fire return intervals in some regions, while increased ignitions have shortened them elsewhere. Introduced pests create novel disturbances native trees have no evolutionary history with. Forests demonstrate remarkable resilience across appropriate timescales. A mature forest might burn completely, killing all trees. Stand in the aftermath, and it looks like total destruction. But return in a decade, and you'll find young trees several feet tall, shrubs providing cover. Return in 50 years, and a new forest has formed, different in species composition perhaps, but functional, cycling nutrients, supporting wildlife, storing carbon. The forest has reset, but it has regenerated. This resilience depends on the integrity of underlying processes, seed sources nearby, soil intact, mycorrhizal networks surviving, appropriate disturbance frequencies. Disrupt these too severely, and resilience fails. Clear-cut a forest, compact soil with heavy machinery, spray herbicides, replant with monocultures. The forest that regrows, if it regrows at all, differs fundamentally from what preceded it. The question facing forests now is whether the changes we're imposing exceed their capacity to respond. Can forests adapt fast enough to track climate change? Can simplified, managed forests maintain the functions that complex natural forests provide? Can we preserve enough old growth to serve as refuges and seed sources? The answers depend partly on timescale. In the short term, years to decades, many forests will decline as they struggle to adapt to rapid change. Species will shift in abundance, some disappearing, others invading. Structure will simplify in some locations, become more complex in others. Function may degrade, carbon storage decreasing, water cycling disrupted. Over longer time scales, centuries to millennia, forests will likely persist in some form, adapted to whatever conditions ultimately stabilize. The forests of the future may contain different species, have different structures, provide different services than today's forests. But forests are ancient. They've survived ice ages, volcanic eruptions, continental drift, asteroid impacts. They've persisted through climate changes as dramatic as the current one, though never quite this rapid. The forest operates across all time scales, from instantaneous propagation of chemical signals to millennial persistence of ancient trees. Understanding forests requires thinking across this enormous temporal range, connecting the immediate to the ancient, recognizing that the forest you see today is simultaneously responding to yesterday's weather and shaped by disturbances from centuries ago, while setting conditions that will determine its composition decades hence. Time in the forest is layered, just like the canopy. Fast processes playing out within slow ones, brief events leaving lasting legacies, ancient patterns persisting through constant change. Draw a line through a forest, a road, a fence, a cleared strip for power lines. The line looks clean on a map, a simple boundary between forest and not forest. But at ground level, where roots intertwine and fungal networks spread, that line fractures systems that took centuries to build. Analysis of global forest cover reveals that 70% of remaining forest sits within 1 kilometer of a forest edge. Most of what we call forest exists in a compromised state, its interior processes infiltrated by conditions from outside. The edge begins where sunlight penetrates. In intact forest, the canopy filters light into diffuse luminescence. At a new edge, full sun strikes the ground directly. Temperatures spike, winds strengthen, humidity drops. Species adapted to stable, shaded interiors suddenly face conditions they evolved to avoid. The physical changes propagate inward. Edge effects can extend up to 1,000 feet into rainforest. Wind penetrates deeper, drying soil and air. Temperature swings grow more extreme. The humid microclimate that buffered against drought dissipates. Trees at the edge, having grown in protected conditions, lack structural reinforcements to withstand constant wind. Many die. This mortality creates a feedback loop. Dead trees open more gaps, allowing deeper penetration of sun and wind, killing more trees. The edge advances inward, consuming forest even without additional clearing. The species composition shifts. Shade-tolerant plants that dominated the interior die back. Opportunistic species, adapted to disturbed conditions, fast-growing, weedy, colonize the edge zone. These invaders often originated outside the forest, arriving as seeds on wind or carried by animals moving between fragments. Lianas, woody vines, proliferate along edges. They grow faster than trees, climbing trunks and draping over canopies, pulling branches down with their weight. Both lianas and fast-growing trees fail to sequester as much carbon as slower-growing species they replace. The forest's carbon storage capacity diminishes. Animal communities unravel through multiple mechanisms. Some species simply cannot survive in small fragments. Their territory is too large. Their population needs too many individuals. Large predators disappear first. When fragments drop below certain size thresholds, the predators vanish through mathematical inevitability. Their absence cascades downward. Prey populations, released from predation pressure, surge. These herbivores then overconsume vegetation, particularly targeting seedlings. Regeneration stalls. The forest ages in place, its canopy slowly dying without replacement. Other animals decline because fragmentation interrupts their movement. Many forest species move seasonally, tracking fruiting events, breeding sites, or favorable conditions. Fragment boundaries block these movements. Animals that once ranged across continuous forest find themselves trapped in patches too small. Pollination networks collapse when fragments become too isolated. Many tropical trees depend on specific pollinators: bees, bats, birds, that travel between flowering individuals. When distance between fragments exceeds pollinator flight range, trees can't reproduce sexually, even if healthy. Populations become reproductively isolated. Genetic diversity declines. Inbreeding increases. The underground networks suffer similar disruption. Edge effects significantly alter temperature, humidity, light, and wind conditions, impacting species composition and community structure. These changes affect mycorrhizal fungi. The fungal community shifts towards species tolerant of drier, hotter conditions, generalists rather than specialists. The hub trees, those massive old individuals anchoring the mycorrhizal network, become vulnerable at fragment edges. Their shallow lateral roots, which connected them to neighbors, get severed by the boundary. Their capacity to exchange resources diminishes. When these hubs eventually die, often from edge-related stress, the network loses its central nodes. Connectivity drops. Seedlings suffer most from network disruption. Seedling density declines significantly from continuous forest to fragments, with edge seedling density increasing toward forest interior. Without robust mycorrhizal support, seedlings struggle to establish, especially in deep shade where they depend on network subsidies. Fire risk amplifies along edges. Edge effects make wildfires more likely and more severe. The drier conditions, increased fuel from dead vegetation, and wind penetration create ideal conditions for fire to enter fragments. In regions where surrounding land gets burned regularly, pastures cleared by ranchers, agricultural waste burned after harvest, fire breaches the forest edge repeatedly. Research spanning multiple biomes demonstrates that habitat fragmentation reduces biodiversity by 13 to 75%. The range reflects variation in fragment size, isolation, and surrounding matrix. Small, isolated fragments surrounded by intensive agriculture lose the most species. Larger fragments connected by corridors to other patches retain more biodiversity. The temporal dimension matters critically. Effects are greatest in the smallest and most isolated fragments, and they magnify with time. A fragment might appear healthy for years after isolation, its tree canopy intact, birds still present. But this represents borrowed time, what ecologists call an extinction debt. Populations no longer viable will continue declining over decades until they finally disappear. The effects of current fragmentation will continue emerging for decades, with extinction debts likely coming due. We cannot judge fragment health by immediate appearance. Full consequences unfold across timescales, matching the lifespan of long-lived trees. Studies found that species diversity, tree size, and structural diversity were significantly reduced in fragmented forest and within forest edge areas compared to continuous forest. The trees in fragments grow smaller. Structure simplifies. Fewer size classes, less variation in canopy height. The size and shape of fragments influences outcomes. Long, narrow fragments have more edge relative to interior than compact, circular fragments. Species diversity and structural diversity are strongly influenced by total size, core area size, and shape of forest fragments. A 100-acre fragment shaped like a ribbon might have almost no true interior. 100 acres in a square configuration retains some protected core. The surrounding matrix, land use adjacent to fragments, determines edge severity. When forest fragments are encircled by pastures regularly burned, fire destroys secondary vegetation and continually reopens fragment edges. Fragments surrounded by secondary forest or shade plantations experience milder edge effects. Some fragments develop novel community compositions through what researchers call landscape divergence. Rather than simply losing species through extinction, fragmentation can promote important landscape scale differences among biotas. Each fragment, isolated and experiencing unique local conditions, develops its own trajectory. Species combinations that never coexisted in continuous forest appear in fragments. Whether these novel ecosystems function well remains uncertain. They might maintain some ecosystem services, carbon storage, soil retention, or they might represent transitional states, slowly degrading toward collapse. Conservation responses focus on maintaining or restoring connectivity. Wildlife corridors, strips of protected habitat linking fragments, allow animal movement between patches. These corridors need not be pristine forest. Even degraded habitat can facilitate movement. The key is continuity. Reforestation between fragments can restore connectivity gradually. Trees planted in cleared areas eventually grow into forest, though the process takes decades. Meanwhile, reforested areas provide stepping stones, patches of habitat allowing gradual movement across landscapes. Buffer zones around fragments can reduce edge penetration. Land use in these buffers might be limited to low-intensity activities: shade-grown coffee, managed secondary forest, agroforestry systems maintaining some tree cover. These buffers won't support interior specialists, but they soften the transition from forest to matrix. But ultimately, preventing fragmentation proves more effective than attempting restoration afterward. Urgent conservation efforts are needed to halt ongoing forest fragmentation, and reforestation efforts are required to reconnect isolated patches. Once networks break, once mycorrhizal connections sever, hub trees die, animal movement patterns disrupt, microclimates shift, repair becomes difficult, expensive, and uncertain. The forest evolved as a connected system, its parts interdependent across scales from microscopic fungal threads to landscape-scale animal migrations. Fragment it, and you break not just physical continuity, but functional relationships. The fragments persist, but diminished, their complexity reduced, their resilience compromised, their capacity to support full diversity gradually eroding as slow consequences of isolation work through the system over decades and centuries. In 1916, ecologist Frederick Clements stood in a Michigan forest and made a radical claim. The forest, he argued, behaved like an organism. It grew, matured, reproduced itself on empty fields, and eventually died. The beech maple community before him displayed integrity and identity as clear as any crow or oak. If organisms self-organize and maintain their boundaries against dissolution, then forests qualified. Clements called them superorganisms. The idea caught fire among biologists of the 1920s. They recognized something their predecessors had missed. Collections of individuals could produce behaviors belonging to the collective, not to any member. A slime mold assembles from scattered cells into a unified body. An ant colony coordinates activities across thousands of workers. Why not forests? A rival ecologist, H.A. Gleason, rejected this thinking, arguing the forest was merely a loose gathering of organisms brought together by circumstance rather than integration. Species came and went, depending on climate and geology. Where Clements saw coordination, Gleason saw coincidence. The debate shaped ecology for decades. Both men marshaled evidence from nature. Along the Pacific Northwest coast, the boundary between seaweed communities and spruce forest forms a sharp line, suggesting two distinct superorganisms. But counterexamples abound. Most forest communities don't defend sharp boundaries. Species infiltrate gradually, compositions shift. Computer models showed that few ecosystems self-organize into lasting coherence. Yet the intuition that forests exhibit organization beyond random assembly refuses to disappear. Modern research revives the superorganism concept, though in more sophisticated form. Forests can show emergent behavior, properties arising from component species growing together that differ from what simple aggregation would predict. Consider what we've traced through the preceding chapters. Trees exchange resources through mycorrhizal networks, warning neighbors of threats and recognizing kin. They communicate through volatile chemicals, priming defenses. They pump water through transpiration, creating rainfall. They cycle nutrients through decomposition networks, capturing and recycling resources. Ecosystem self-regulation emerges from dynamic interactions of living and non-living components, with feedback loops maintaining stability without external control. When herbivores increase, predators respond by consuming more prey. When nutrients wash from the system, decomposers work harder. When drought stresses the community, trees close stomata. These feedback loops operate at multiple scales simultaneously. A tree responds to water status in seconds. The mycorrhizal network redistributes resources over hours to days. Species composition shifts across years to decades. The entire forest reorganizes over centuries following disturbances. Resilience, the capacity to absorb disturbance and reorganize while retaining essential function, emerges as a property of the whole system rather than any component. A diverse forest withstands pest outbreak better than monoculture because multiple species provide functional redundancy. When one species declines, others fill its ecological role. But calling the forest a superorganism raises immediate problems. Communities lack centralized genomes and cannot reproduce to perpetuate identity. The forest doesn't evolve as a unit. Its component species evolve individually. The apparent coordination emerges from individual selection acting on separate organisms. This distinction matters. An organism's cells share genetic interest. They propagate or perish together. A forest's trees compete genetically. Cooperation occurs, but only when it serves individual fitness. The mycorrhizal network distributes resources, but fungi and trees both manipulate the exchange. The appearance of altruism often masks underlying competition. Yet emergent properties genuinely exist. Forest structural diversity interacts with disturbance regimes to create novel ecological properties, developing non-linearly. A forest with complex vertical structure catches more light at various angles throughout the day, increasing total photosynthesis. Multi-aged stands recover from disturbance faster than even-aged stands because diverse age classes provide resilience. The nutrient cycling we explored exemplifies emergence. No individual decomposer evolved to cycle nutrients for the forest's benefit. Each pursues its own metabolism, but their collective activity creates stable nutrient availability. The forest benefits, plants grow better, and this growth produces more organic matter. A feedback loop stabilizes the pattern. Similarly, water cycling shows emergent regulation. Trees transpire to facilitate photosynthesis, not to make rain, but millions of trees simultaneously inject enough moisture into atmosphere to trigger precipitation. That rain falls back on the forest. The system self-reinforces. Forests create conditions favorable for forests. temperature regulation works identically. Forests create cooler, more humid microclimates than surrounding areas because trees transpire, shading ground and releasing moisture. Each tree benefits from stable conditions. The forest doesn't regulate temperature for the good of the community. Stable conditions emerge from individual trees, modifying their environment in ways that create collective benefits. Climate regulation represents an emergent property at large scales, with forests absorbing carbon dioxide and influencing atmospheric composition. No tree evolved to regulate global climate, but collectively, forests sequester enough carbon to measurably affect atmospheric concentration. This affects climate, which affects forest growth, creating another feedback loop. The question becomes whether these emergent properties suffice to justify the superorganism label. If superorganism means tight integration, comparable to multicellular organisms, then forests fall short. Their couplings are loose, their boundaries permeable. But if superorganism means a system exhibiting properties emerging from component interactions, properties unpredictable from studying components in isolation, then forests qualify. The debate reflects deeper tensions in ecology between reductionist and holistic approaches. Reductionists argue we should understand forests by understanding components, individual organisms pursuing fitness. Holists argue the forest possesses properties that only appear at system level. Neither purely reductionist nor holistic approaches suffice. Perhaps the most useful perspective treats the forest as operating across organizational levels simultaneously. At the individual level, trees compete for resources. At the population level, groups of related individuals may experience kin selection, favoring cooperation. At the community level, species interact through competition, predation, mutualism. At the ecosystem level, energy and nutrient flows create patterns of productivity and cycling. Each level exhibits its own dynamics and generates its own properties. The forest is simultaneously a battleground of individual competition and a coordinated system cycling resources. Both perspectives contain truth. The challenge is integrating them, understanding how individual selection creates community-level patterns, how ecosystem properties feed back to shape individual fitness. The practical importance exceeds philosophical interest. If forests self-regulate through emergent properties, then preserving those properties requires maintaining conditions that generate them. Fragmentation disrupts communication networks. Simplified structure reduces functional diversity. Removing species eliminates redundancy. These impacts degrade the forest's capacity for self-regulation, making it more vulnerable to disturbance. Management implications follow. If we view forests as collections of individual trees, we might manage them tree by tree. But if forests are systems with emergent properties, then management must consider system-level effects, maintaining mycorrhizal networks, preserving species diversity, protecting hub trees, allowing natural disturbance regimes. These actions preserve processes that generate forest function. Climate change tests these systems in real time. Forests evolved their self-regulating properties under conditions that persisted for millennia. Now those conditions shift rapidly. The feedback loops that stabilized forests may destabilize instead. Drought reduces transpiration, which reduces rainfall, increasing drought. The self-reinforcing cycles that built forests could accelerate their collapse. Yet forests demonstrate remarkable capacity to reorganize. After disturbance, they don't return to exact prior states, but establish new configurations that function in changed conditions. This adaptive capacity suggests resilience beyond simple homeostasis. The forest doesn't just resist change, it absorbs change, reorganizes around it, finds new equilibria. Whether this flexibility suffices for the pace of current change remains uncertain. Stand in a forest, and you stand within a system organized across scales from molecular to continental, from seconds to millennia. The trees around you exchange carbon and information underground through fungal networks. They communicate warnings through airborne chemicals. They pump water from soil to atmosphere, generating rain. They host thousands of species in their canopy layers. They cycle nutrients through decomposer networks. They synchronize reproduction across populations. They create microclimates that buffer extremes. None of this requires consciousness or intention. The forest doesn't decide to regulate itself. The properties emerge from countless individual actions that happen to create stable patterns when viewed at larger scales. But the result, a self-organizing, self-maintaining system exhibiting coordination across space and time, resembles in important ways the integration we see in organisms. Perhaps the forest is best understood not as a superorganism, but as a complex adaptive system, a network of components interacting through feedback loops that generate emergent properties and enable self-organization. The label matters less than recognizing the forest's systematic nature, its integration despite apparent disorder, its capacity to maintain function through constantly changing composition. The secret life of forests ultimately reveals that forests are not just places where trees grow. They are systems, networks, communities, ecosystems. Whatever term we choose, they represent organization beyond the sum of parts. The phenomena we have explored, from the dawn choruses to the decomposer economies, all of them connect, interact, and reinforce each other, creating something greater than scattered trees on soil. But understanding remains incomplete. The forest holds more secrets than we've uncovered, more complexity than we can map, more connections than we can trace. Each answered question reveals deeper mysteries. The forest's true nature remains partially hidden, not because we lack tools to study it, but because its essence lies in relationships too numerous and subtle to fully comprehend. A web of life, so finely woven, so deeply interconnected, that we've only begun to glimpse its pattern.